The term calico refers to the coat color variations. Calico cats is a cat with tricolor coat, orange, black, and white. Calico cats are all female, and the sex of the cats result in the tricolor formation and not the other way around. Thus, there are very rare chance for male to have this coat pattern. This phenomenon can be explained through the understanding of X-chromosome inactivation. X-chromosome inactivation is a mechanism to counteract the unequal dosage composition of X-linked genes in female and male organisms by silence or inactivate one of the two X chromosomes in female. Compared to the gene pair Y chromosome, the X chromosome contains many genes that is essential for the development and survivability. In female, there are two X chromosomes but only one in male. And animal development is very sensitive to imbalance in the number of genes, so extra copies of genes could lead to abnormal phenotype expression and even can be lethal. The process of X-inactivation in calico cats can be observed in Human X-inactivation Center, ZIC. ZIC located at the Q arm of the X chromosome, and within the ZIC, there is X-inactive specific transcript gene, which only expressed on the inactive X chromosome but not on the active X chromosome. X-inactivation occurs during the early development of the embryo. Thus, depending on the initial inactivation, all the daughter cells and tissues will continue to have the same pattern of X chromosome inactivation. So how the X chromosome inactivates itself? In each cell, one of the two X chromosomes will undergo Lyonization, and this process occurs randomly. On the inactive X chromosome, this gene will be expressed. So when there are two X chromosomes in a cell, the inactive X will express the ZIS gene, while the active X will not express the ZIS gene. This results in the production of 17 kb RNA in the inactive X. This RNA is not like other RNA because it doesn't code for any proteins. Through the fluorescent in situ hybridization, it was found that the transcript of the ZIS concentrates at the condensed X-inactive chromosome, or also known as the bar body. As you can see here in this figure, this is because the RNA will remain in the nucleus and surrounds the cluster of X-inactive chromosome. So from that observation, researchers hypothesized that the ZIS RNA could act as a barrier around the X chromosome, preventing the binding of RNA polymerase to initiate normal transcription. Second, this RNA could work individually or with other nuclear factors to cause changes in the chromosome, which then silence the chromosome. Third, this transcript could cause the chromosome to become non-functional by promotes the binding of X chromosome to a specific site on the nuclear membrane. And lastly, the ZIS RNA itself is the switch that causes the silence of the X chromosome. Back to the calico cats. The X chromosome carried the coat color. There are three possible combinations for coat color in female cats. C, D, E in this figure. So because ionization is random in each cell, it can be predicted which one of the two X chromosomes will be inactivated. In one cell, it can be the chromosome with black allele to be inactivated, while in another cell, it can be chromosome with orange allele to be inactivated. Hence, only the female are capable of expressing the mosaic expression of coat color. Thank you.